Hello and welcome to G Fun Facts Online, the show that unpacks the incredible science shaping our world. Today, we're diving deep into one of humanity's biggest, most visible and most persistent headaches, plastic. Just look around you right now. Chances are, you can spot at least 10 things made of plastic without even trying. It's in our homes, our cars, our clothes, and unfortunately in our oceans, our soil, and even inside our own bodies. Every single year, humanity produces over 400 million tons of new plastic. To put that in perspective, that is the equivalent weight of the entire human population. Imagine a clone of every single person on Earth, but made of plastic, created every single year. That's the scale we're talking about. We've been told for decades that the answer is recycling, to dutifully put our plastic waste in the right colored bin. But what if I told you that recycling, as we know it, is a temporary fix at best? And what if I told you that the real, long-term solution to this gigantic, synthetic problem might be microscopic? That scientists are now breeding and training a tiny army of specialized microbes to not just eat our mountains of plastic waste, but to transform it into things we desperately need, from life-saving medicines to brand new, eco-friendly materials. Stick around, because today we're exploring the revolutionary science of biorecovery, and we're meeting the incredible plastic-eating microbes that could save the world. Let's get started. To truly appreciate the solution, we first have to understand the sheer, overwhelming scale of the problem. As I said, 400 million tons a year. A huge portion of that is designed for a single use. The bottle you drink from, the wrapper on your lunch, the bag you carry your groceries in. After a few minutes of convenience, it's tossed away, and we hope it goes into the recycling system. But the reality of what happens next is far more complex and far less effective than we'd like to believe. The most common method is called mechanical recycling. You take a plastic bottle, you shred it into tiny flakes, you wash it, you melt it down, and you form it into something new. The fundamental issue with this process is that plastic is made of long chains of molecules called polymers. Think of them like long strands of spaghetti. That's what gives plastic its strength and flexibility. But every time you melt and reform it, those strands get shorter and weaker. It's like making a photocopy of a photocopy of a photocopy. Each time, the quality degrades. A pristine water bottle can't be recycled into another pristine water bottle forever. Soon, it has to be downcycled into something of lower quality, like fibers for a carpet, or plastic lumber for a park bench. And after that, its next, and final, stop is the landfill. Then, there's the sorting nightmare. There are many different types of plastic, each with a different chemical recipe. You've seen the little numbers in the triangles: PIT, HDPE, PVC, and so on. They are fundamentally different materials. You can't just melt them all down together any more than you can bake a cake by throwing flour, eggs, motor oil, and sand into a bowl. The result is a useless, brittle mess. So recycling plants have to use complex, expensive systems to try and sort this jumble of plastic waste. It's a difficult, imperfect process. All of this means that globally, only about 9% of plastic waste is actually recycled. The rest, the vast majority, piles up in landfills, is incinerated, or worse, escapes into the environment. It clogs our rivers, forms massive garbage patches in our oceans, and breaks down into insidious microplastics that get into our food chain and our water supply. For decades, it has felt like an unwinnable battle, a mountain of our own making that just keeps growing, with no peak in sight. But science, as it so often does, found a new path forward in a very unexpected place. The story of our tiny, plastic-eating heroes begins in 2016. Imagine a team of researchers at a plastic bottle recycling facility in the city of Sakai, Japan. They weren't looking at the big machinery. They were looking at the gunk, sifting through the sludge, the wastewater, and the sediment of the plant. They were on a biological treasure hunt, searching for any form of life that might have evolved to take advantage of this new, abundant, man-made buffet, and they found it. They isolated a brand new species of bacteria, one never seen before. They gave it a fitting name, Noxidianella sacaiensis. This microbe wasn't just surviving in a plastic-rich world, it was dining on it. It was using PET plastic, the very material that makes up our water and soda bottles, as its primary source of energy. This was a monumental discovery. For years, scientists knew that some fungi and bacteria could slowly, almost imperceptibly, nibble away at plastic, but it was a process that took hundreds of years. It was more of a geological process than a biological one. Ideanella sacaiensis was different. It had evolved a specialized two-enzyme system. You can think of these enzymes as a molecular knife and fork. The first enzyme, which scientists fittingly named PETase, acts like a knife. It attacks the long, tough polymer chains of the plastic, chopping them up into smaller, more manageable pieces. Then, a second enzyme comes in like a fork, breaking those smaller pieces down even further, into their most fundamental chemical building blocks, terephthalic acid and ethylene glycol. For the bacterium, this is just lunch, a good source of carbon to live and reproduce. For the scientific community, it was a thunderclap. It was the blueprint. It proved that nature, given enough time and the right conditions, could evolve to solve our pollution problems for us. This discovery threw open the floodgates of research. Scientists started looking everywhere. In soil, in compost piles, in wastewater treatment plants, even in the guts of insects like the waxworm, which is infamous for being able to chew through plastic beehives. And they found more plastic eaters. They found fungi and other common bacteria, like Comomonas testosterone, that also had an appetite for plastic components. Nature had provided the proof of concept. Now it was time for humanity to get into the driver's seat and hit the accelerator. Finding these naturally occurring plastic eaters was like discovering a person who could jog a marathon. It's impressive, for sure. But to tackle the global plastic crisis, we don't need a few joggers. We need an army of Olympic sprinters. This is where the incredible fields of synthetic biology and metabolic engineering come into play. In simple terms, these disciplines are about editing and reprogramming the DNA of living organisms to give them new and useful abilities. Scientists are essentially taking these natural microbes and sending them to a super soldier training program. One of the primary strategies is enzyme engineering. The PETase enzyme from our Japanese bacterium is a great start, but it's a bit slow and it's a bit of a diva. It works best at a comfortable room temperature. The problem is, plastic, especially its tough crystalline parts, breaks down much more easily when it's hot. So researchers are using powerful computers and artificial intelligence to study the precise 3D structure of these enzymes. They're figuring out exactly how they latch onto the plastic and make their cuts. Then, they make targeted changes to their genetic code to create new, improved versions. They're creating mutant enzymes that are far more stable at higher temperatures and dramatically more efficient. In one stunning experiment, scientists took the PETase enzyme and combined its DNA with the DNA from a different enzyme, one found in a compost-dwelling bacterium that's used to breaking down the waxy cuticle of plants. The result was a hybrid, a super-enzyme, that could digest PET plastic up to 6 times faster than the original. But why stop at just making them faster? The next level is called metabolic pathway reconstruction. Think of a bacterium's metabolism as a huge, complex chemical factory with thousands of different assembly lines or pathways, all running at once. Scientists can go in and act as factory foremen. They can rewire these pathways. They can tweak the genetic code to make the bacteria not only break down plastic into its basic parts, but then take those parts and shuttle them over to a completely different assembly line to build something new. This clever trick is often called biological funneling. You can take a messy, mixed stream of different kinds of plastic waste, break it all down into its basic carbon atoms, and then funnel all that carbon towards the production of a single, pure, high-value product. It's the ultimate in intelligent recycling. To make all of this even more effective, Scientists at Rice University tackled a very practical problem. How do you get the bacteria to actually stick to the smooth, slippery surface of plastic? They found inspiration in the sea. They looked at mussels, which are the undisputed champions of sticking to wet, slippery surfaces. They identified the gene responsible for the mussel's powerful adhesive protein and engineered it into their plastic-eating bacteria. The result? Sticky bacteria that could latch firmly onto pet surfaces, dramatically increasing the speed and efficiency of degradation. It's like giving your microscopic cleanup crew their own set of high-tech grappling hooks. And this is where the story truly transcends from simply problem-solving into the realm of the genuinely mind-blowing. The ultimate goal here isn't just to make our plastic waste disappear. That would be a huge win, of course, but it's not the full potential. The true beauty of biorecovery lies in upcycling. The process of turning low-value trash into high-value, desperately needed resources. Remember those building blocks the bacteria create, terephthalic acid and ethylene glycol? Those are useful chemicals on their own. But by re-engineering the microbes' internal factories, we can have them produce an absolutely stunning array of products. For instance, they can make brand new bioplastics. We can feed a bioreactor old, discarded PET bottles and have engineered bacteria consume them and produce something called PHAs, or polyhydroxyalkanoates. PHAs are a family of plastics made by microbes, and they are fully biodegradable. Some are even biocompatible, meaning they can be used for medical implants that safely dissolve in the body over time. This creates the perfect closed-loop system. Old, polluting plastic is transformed into new, environmentally friendly plastic. It's a cycle of creation, not just destruction. But hold on, because it gets even more amazing. In a landmark breakthrough at the University of Manchester, scientists engineered a bacterium called Pseudomonas putida to perform an incredible feat. They fed it the chemical components derived from plastic waste, and the bacteria's rewired metabolism produced human therapeutics. I am talking about complex, vital proteins like insulin, which millions of diabetics rely on every day, and interferon, a powerful drug used to treat cancers and viral infections. Let that sink in for a moment. We are on the verge of being able to turn a landfill of discarded plastic bottles into a source of life-saving medicine. This could dramatically lower the cost of these drugs and make them accessible to more people around the world. The list of possibilities goes on. These monomer building blocks can be used to synthesize a whole range of fine chemicals, like the aromatics used in perfumes and fragrances, or special types of alcohols used as industrial solvents. Researchers are also figuring out how to make things like nanocellulose, a super strong, lightweight material with applications in electronics and composites, and rhamnolipids, which are powerful, natural soaps or surfactants. All of this, from a starting material that we currently pay to bury in the ground. This isn't just recycling. This is modern alchemy. It's transforming a global curse into a treasure trove of resources, and it is the absolute foundation of a truly circular economy, where the concept of waste simply ceases to exist. Now, as thrilling as all of this is, we have to be realistic. We are not going to be releasing swarms of super sticky, genetically engineered bacteria into the Great Pacific Garbage Patch tomorrow. There are still some significant hurdles to clear before this technology can be deployed on the massive industrial scale needed to make a dent in our 400 million ton a year problem. First, there's still the efficiency issue. While the engineered super-enzymes are much faster, we need to continue to improve their speed and robustness to handle the sheer volume of waste economically. Many plastics also contain highly crystalline regions, which are like tightly packed molecular fortresses that are very difficult for the enzymes to break into. Scaling up is the other massive challenge. A successful experiment in a 1-liter flask in a lab is one thing. Building and operating a bioreactor the size of a building, one that can process thousands of tons of mixed, dirty, real-world plastic waste from a city's recycling stream, is a whole different ballgame. These processes need to be energy efficient, and have a low carbon footprint to be truly sustainable. But the global scientific community is tackling these challenges head-on with incredible ingenuity. They are developing hybrid approaches, where chemical or mechanical pretreatments are used to rough up the plastic first, making it more accessible for the microbes to get to work. They're using advanced, high-throughput screening methods and analyzing the DNA of entire ecosystems, a field called metagenomics, to rapidly discover new and even more powerful enzymes hidden in the natural world. Artificial intelligence is playing a critical role, using machine learning to model and predict how changes to an enzyme structure will affect its function, dramatically speeding up the design of next-generation super-enzymes. And perhaps the most futuristic idea of all is the concept of living materials. Scientists are experimenting with creating plastics that have the dormant spores of these engineered bacteria embedded directly within them from the start. When the product reaches the end of its life, say when your plastic container cracks, you could activate the spores with a specific signal, perhaps a quick blast of heat or a spray of a nutrient solution. The bacteria would then wake up and begin digesting the product from the inside out. Your waste would literally eat itself. So where does this all leave us? Our planet is facing an unprecedented plastic crisis. For decades, our only strategy has been a flawed and inefficient take, make, dispose model, with a tiny bit of downcycling on the side. But we are now standing at the threshold of a new era. An era powered by the smallest and most ancient forms of life on our planet. The chance discovery of Ideonella sakayensis in a Japanese recycling plant, combined with the explosive power of synthetic biology, has given us a new and powerful set of tools. We are finally learning to work with nature, to augment its genius, and to direct its incredible chemistry towards solving our most pressing, man-made problems. The road from today's laboratories to a full-scale global, microbial-powered circular economy is still a long one. It will require immense investment, brilliant research, and bold innovation. But for the first time in the history of plastic, we have a clear, sustainable, and genuinely hopeful path forward. A path where a mountain of plastic waste is no longer seen as a blight, but as a valuable resource, a feedstock for medicine, for new materials, for a cleaner, healthier, and more prosperous world. The solution to one of our biggest problems might just be the smallest thing you can possibly imagine. Thanks for tuning in to Fun Facts Online. Join us next time as we continue to explore the amazing science that is shaping our future.